Good afternoon and welcome to the Cancer 2.0 session. We're really excited. We've got lots of people we want to introduce you to today, experts in their field, and we've got a lot that we want to tell you about what GEL is up to within this field. My name is Emma Mccargo. I am the Cancer Programme Lead for Genomics England, and I want to introduce you to Parker Moss. He's our commercial officer, chief commercial officer, I should say, and our research tribe lead. Parker.
Thank you very much, Emma. So the first thing I want to say about cancer is how incredibly proud and excited we are to be collaborating with the NHS and the Genomic Medicine Service that the NHS have rolled out across the country. So for those of you who didn't hear what Dame Sue Hill said earlier about what we're doing in cancer today, it really is world leading. So for the last several months, we have been sequencing, whole genome sequencing across the country, all children with pediatric cancers, all sarcoma patients, and all acute leukemia patients. And we've just had approved and will be introducing in the coming months whole genome sequencing of triple-negative breast cancer patients, of ovarian cancer patients, high-grade serous, of the other hematological cancers. We'll be extending children to include young people, and we're going to be sequencing pan-tumor across metastatic and refractory conditions that are not well managed by standard of care. So this really is a world first. It's not just the number of indications that we're covering, but it's also that the NHS have managed to successfully set up the infrastructure, the clinical governance, the clinical buy-in, and the interpretation capability to roll this out at a national scale. And it's a tremendous thing for patients with cancer in the UK. So that's absolutely thrilling.
So, Paco, I wondered if you could tell a little bit more about what our vision is from a clinical, but also from a research point of view.
Yes, okay, so I'll start clinically. So cancer has been called the emperor of the maladies, and the reason for that is that it is an exquisitely complex disease. So cancer is multigenic, it's heterogeneous, it's metastatic. Cancer interacts with the germline, but it also interacts with the immunome, and increasingly it appears that it's interacting with the microbiome. So it really is a complicated disease to diagnose, to prognose, and to plan treatment for. And so what we want to make sure for the cancer patients that receive whole genomes is that we're taking the most comprehensive view of the genome to make the right treatment decision for them. And it's not just about the detail, it's also about a different way of looking at cancer. So because of this complexity that I laid out, it's very difficult for clinicians to be hypothesis-led, to start from the phenotype and make their treatment decision or their treatment planning from that basis. So we are engineering a system in collaboration with the NHS, which is hypothesis-free. We're really unlocking the genome and allowing the genome to tell us the story about the patient's cancer. And we think that's going to introduce really exciting clinical trial opportunities for patients, particularly in a recurrent or a refractory setting. It's going to give us biomarkers that we can follow to track minimal residual disease along their journey post-remission. And there really will be a growing body of evidence that patients are going to benefit clinically. But getting the clinical service right is not just about accuracy and consistency. It's also about getting the operational details right. So, when the first genome was sequenced at the beginning of this millennium, it cost over $1 billion, and famously it took many years to sequence that patient. Now sequencing can happen in less than a day, but the sequencing is just a part of the story. The whole end-to-end journey involves taking tissue from the patient, extracting the DNA, sending that to a central sequencing lab, running it through our bioinformatic pipelines, through decision support platforms, back through genomic lab hubs who combine other modalities like immunohistochemistry and imaging, and then back to genomic tumor board. So it's a long end-to-end process that takes several weeks. And we know that patients in England deserve better than that, so we're working very hard looking at new technologies, and you'll hear an expert panel talking about nanopore sequencing, which has a different form factor that will allow sequencing to happen much closer to the patient in the hospital, and we hope that that will demonstrate turnaround time benefits, and that we can eventually get sequences to be turned around in a week or a little bit more than that.
Yeah, it's wonderful, isn't it, for the patient? What's the view, and where are we going from a research point of view?
Yeah, so from a research point of view, this is where I have to say how grateful we are to all of our participants. So we've sequenced 17,000 or more cancer patients as part of the 100,000 Genome Project, and now we're prospectively sequencing patients in the NHS. And today, prospectively, over 95% of those participants consent to allow us to have access to their data for research. So that's a tremendous thing. And what does that mean? That means we collect all of their clinical data retrospectively and prospectively, and you're going to hear in our digital pathology panel that we're also going to be collecting their images, radiology and pathology images. We have their full text pathology and radiology reports, their cancer regimen data, and that's through partnership with NHS Digital and Public Health England. And we have the ability to not just rephenotype those patients, but also to recruit them into clinical trials and to resample them to get precious human tissue for these functional genomic pipelines that can try to convert our data on associations between genotype and disease to actual causal relationships. So it's a data set that is a very, very powerful resource. We put all of our data sets, all of our data into Amazon Web Services today, and that's fronted by a terrific bioinformatic platform, which is run by a company called LifeBit, which is a great British startup that just was tremendously successful in a fundraise a couple of weeks ago. And that creates a really nice research environment, which is extremely secure and trusted, that researchers from around the world can come and look at. So today we have 4,000 academics, and some of the best ones are in the room with me today. And we have 35 commercial partners, and of those commercial partners, we have 8 of the top 10 pharma companies in the world who are researching our data and who are learning how to build better drugs, better diagnostic tools, who are doing these preclinical studies to prove causality, which de-risks clinical drug discovery and clinical development pipelines, and we're building real-world evidence tools as well, so that we can really demonstrate that the effects that drugs are meant to generate in very carefully managed clinical trial settings actually have that same effectiveness in the real world. So some of the strategies that we're doing to improve the experience and improve the quality of data for our research community is, number one, we're opening up our research pipelines to researchers who are working in the machine learning space. And you're going to hear later from Serena some of the work she's doing in homologous recombination and mismatch repair to introduce the absolute cutting edge algorithms that can look for markers of genomic instability. And we'll be incorporating those in our clinically accredited NHS pipelines. So that's one strategy. And another thing we're doing is we're going to be adding digital images of pathology and radiology into our data sets so that researchers can look at not just the molecular, but also the spatial context of solid tumors. So I think we're creating a really powerful world-leading research resource as well.
Wonderful. That's wonderful. So maybe I would hand over to...
Yes, let's call on the first panel.
To the first panel. Yeah.
Thank you. So I have on the stage with me today some of the real experts in sequencing in the wet lab, and I'm going to ask both of them to introduce themselves. Andy, please.
Hi, I'm Andrew Beggs, and I'm a professor of cancer genetics and surgery at the University of Birmingham.
And Greg.
My name is Greg Elgar. I'm Director of Sequencing Research and Development at Genomics England.
Thank you. So guys, we have been working for many years very successfully with the gold standard short-read sequencing, which produces very reproducible and exquisitely accurate results. But we're now beginning to look at long-read sequencing, specifically with nanopore, so nanopore sequencing for long reads and methylation. And maybe I can ask both of you, I'll start with you, Andy, why are we doing that now? What are some of the benefits?
I see long-read sequencing as an evolution on existing techniques. It's complementary, but an evolution. And it allows us to easily identify things that short reads may have struggled to do in the past, things like structural variants, and end-track fusion is an example of that on DNA, but also turnaround time, speed, ease of accessibility. And I think the final thing that's most important is methylation. And we now know from several Nature and Nature Medicine papers that methylation could be used as a biomarker for deciding, if, for instance, in brain cancer, what the subtype of brain cancer is, which has profound implications for treatment.
Thank you. Greg, same question to you.
Yeah, I guess I'd look at this from a more operational standpoint. So short-read sequencing has been very much the state of the art for a number of years now. But long-read sequencing has been at the fringe, and Oxford Nanopore have really led in the UK, the development of instruments and the technology itself. And now is the time, I think, that they are starting to look as if they have clinical grade applications and clinical grade instruments that could be used in future healthcare. So I think it's a really good time now. I think we really have to jump on the back of that and push the technology forward and work with clinicians, with researchers. We need to bring academic researchers together with clinicians, and GEL is a great way to do that. Genomics England is good at facilitating that. So I think that's what we need to do. We need to do it now. We need to try and get it into the healthcare setting in the next year or two.
Thank you. And Greg, having had a quick look at the audience that are joining this call, there are quite a lot of clinical scientists and technical people in the room. So maybe you could just go into a little bit more detail. What has changed now in terms of quality and throughput that makes nanopore sequencing kind of ready for prime time?
Yeah, I mean, there are a number of things we have to consider. First of all, the economics of it. So long-read sequencing has been very expensive. Nanopore have driven down the costs, especially at scale, and I think it's now competitive with short-read sequencing, and we mustn't forget that that's a serious consideration. But at the same time, they have made technological advances that have made the sequencing more accurate. They have enabled us to sequence a whole genome on one of their flow cells, so the capacity of the sequencing has increased. And they've introduced instruments in the last year or two that can really match any of the short-read instruments in terms of scale and capacity. So the current instrument that we're using, the PromethION, in our lab, can sequence 48 genomes simultaneously, and we can get really good data from that machine in about 24 hours for those 48 genomes. And that's really up there with the capacity of other technologies.
Okay, so I think it's just such an exciting opportunity to introduce this complementary technology into the lab and ultimately into the clinic. And so maybe, Greg, you can tell me, Andy, you can tell me a few kind of patient examples of where you feel this might make the difference to patients in the clinic.
I think before just the patients, the speed of being able to get the data to a patient is fantastically important with long-read sequencing technologies. So an example is in brain cancer. It's very important to be able to get a rapid clinical diagnosis, both in adults and children. And one of the advantages of long-read sequencing, you can provide the structural variants, the copy number variants, the methylation, most importantly, to accurately subtype a patient's tumour and take it forward to a diagnosis so you can pick the right treatment. Another example is sarcoma and hematological malignancies, but also it could be pan-cancer. So, for instance, NTRK fusions, larotrectinib has been licensed now for NTRK fusions across the whole spectrum of cancers. And one of the advantages of long-read sequencing is we don't need to turn to RNA and RNA probe bases. We can go straight from DNA using long reads. And that means you can wrap everything up into a very neat package with a single assay.
Okay, I mean, that's extraordinarily exciting. And maybe as you run a lab, as you both run labs actually yourself, you can tell us a little bit about the footprint and the form factor of these new devices and how that might change things operationally.
I mean, one of the great things about the long-read sequencing, the nanopore devices exist. They're very portable. You have the smallest one, the MinArm, which is about the size of a big USB stick. But the PromethION, which is what we both use for clinical grade type sequencing, I have literally picked it up and put it under my arm. And it is durable and robust. And it makes it a lot easier to take this type of sequencing to the patient, which in the past, the sequencing has had to go to the machine rather than be a bit nearer to the patient. And that allows us to speed it up and really democratize it, widen access and participation for all patients.
Yeah. Greg, we're in the middle of a kind of validation program in which you're testing against the gold standard. Perhaps you could explain a little bit about that program and some of the challenges that we're facing operationally and technically and with the infrastructure to make this live and real.
Yeah, absolutely. So the backbone of a pipeline is similar to that of short-read sequencing, but the challenges are more significant in certain areas. So the extraction of DNA from high-quality tissue presents two problems. One is getting the high-quality tissue in the first place, and we need to work very closely with surgeons, pathologists, cancer clinicians in order to expedite the movement of that tissue to a central lab where the DNA can be extracted, or it needs to be done locally to a high-quality level. And once that's moved into the lab, the sequencing part now actually has become pretty straightforward. The machines are more reliable and more robust, and we have a pretty low failure rate if we get reasonable quality DNA in the lab for long-read sequencing now, and that's really important. Then there are also some challenges in terms of interpretation, because I think certainly at the outset, we would probably be looking at using short-read sequencing and long-read sequencing alongside each other, because they both have their strengths and weaknesses. And so it really enriches the data that you can get from a whole genome when you use long and short-read sequencing together. But we haven't really worked out the best way of actually merging those two sets of data. And there are various ways we can do it. And then leading on from that, we then need to have clear interpretation that we can then hand back to clinicians. And that's also an area where we have to really work things out. So we're going to get long-read data, short-read data, methylation data, and we need to put that together so it doesn't conflict, so it actually supports each other, because they are in a way they're validating technologies. So long-read sequencing and short-read sequencing are done completely differently, are using completely different methodology and technology. And so if you find a mutation in both, then you've immediately validated, or each validates the other. So there's some real advantages, but we need to work that out. It's also a really rapidly evolving technology still. And so in order to stick to a specific end-to-end pipeline, we need to make really important decisions about what we take on board and what we leave behind.
Yes, this is a real challenge, isn't it? Of course, in the scientific method, you want a stable baseline, but the baseline is shifting every month. Right. There's the quality of pores and the chemistry, but also the quality of the machine learning algorithms for base calling improve. And so that must be a very challenging thing to do. And we've talked a lot about the long read, but you've both mentioned methylation. Andy, maybe you can tell me a little bit more about the clinical application of methylation and whether you really think that, that nanopore sequencing could be a substitute to orthogonal methylation testing.
So my laboratory has undertaken quite a lot of methylation-based testing recently using nanopore, and to be honest, it's a killer application for the technology. It beats everything currently going, all the old techniques using short-read sequencing and other technologies like pyrosequencing. We see base accuracies comparable to whole genome bisulfite sequencing. And so the advantage of that is we can use that very rapidly and validate it very rapidly into clinical practice. Certainly it's transformational for us. I mean, one of the other exciting things is, for the technical minded on this discussion, is that we can phase methylation as well. So we can actually work out the maternal and paternal contributions within the cells and various things. And that is fantastically interesting. It's something we've never been able to do before. So it's unique to long reads, and it's mind-blowing in terms of what it can open up for us in understanding how these cancers work.
And in terms of this question, and for the less technically minded on the call, maybe both of you can explain how the principle of using a pore for sequencing differs from sequencing by synthesis and chopping up the genome, and why, in some ways, that how that affects our view of ground truth and the operations.
Well, it's very clever. It's basically a protein pore in a membrane, and you detect a voltage as the DNA strand goes through it for nanopore sequencing. And that has advantages and disadvantages. The advantages is you can stick anything through it, not just DNA. Theoretically, protein can go through it as well. And from the voltage change, you can understand whether a base has been methylated or unmethylated. With short-read sequencing, it works by adding coloured nucleotides onto a flow cell that shine when the laser shone on them. And that is very good and very accurate, but it doesn't allow you to get access to things like methylation.
Yeah, fascinating.
Yeah, I mean, and the real reason for that is you are sequencing the real genome with nanopore technology. You know, it's the DNA that's come out the cell is going straight through that pore and being read. Whereas with other technologies, quite often you're reading secondary copies of DNA. So you lose a lot of that really vital information that could be really key, for instance, methylation, but other modifications as well. DNA carries lots of other modifications besides just 4 bases, and potentially, if you're reading the native DNA molecule, you can read any of that information.
So isn't it exciting that we have these new players in this sequencing ecosystem, and not just on the infrastructure side, but also on the software side? I'm aware that we're evaluating various variant callers at the moment, and from what I understand, both of you are working closely with Google and their Pepper deep variant algorithm, which uses deep learning to actually take a visual impression of the genome. Who would have ever thought that Google would have been one of the leaders in variant calling in cancer sequencing? Maybe you can tell me a little bit bit about what you're doing with them, Andy.
Yeah, so it's an amazing approach. It completely turns on its head variant calling that we've seen in the past. In the past, you use mathematical approaches to match the sequence you get off the sequencer to the reference human genome. But what Google have done, have turned that on its head and turned it into a human method. Essentially, it looks at the genome and it puts it as a picture, and you see a map of both the genome and the sequence genome, and the machine learning algorithm works out where it should go, but like the human eye would. So it's completely different from any mathematical approach, but it has the advantage of being incredibly accurate. They've taken how a human would do it, sped it up, and used machine learning to leverage the accuracy. And so we've seen substantial increases in accuracy using the Pepper Deep variant algorithm within our own laboratory, and I know that there's further to go with it.
Anything on variant calling or base calling?
Yeah, I would probably add that this is, again, a rapidly evolving field. And it is great to see completely different players using quite really novel approaches. And it's partly because of the way the signal comes off the machine. It's completely different to the somewhat synthetic approach of other methods. It is the real raw signal, and that gives you the opportunity to play with it. And it also has given Genomics England the opportunity to really engage with top academic researchers as well, who are really keen to develop algorithms to read these sequence traces better. So it's a really exciting collaboration for Genomics England, for clinicians, for academics. It pulls everyone together, and I think that's, it really makes me working in GEL feel like a real enabler of this kind of work, and it's great fun.
So we have one question from the floor, and I think this question highlights how technical our audience is. So you can choose who takes this one. So here's the question. I thought methylation was stripped before the gametes were formed, so how can it be phased?
Good question.
So DNA is methylated all through our life. So yes, there is a stripping and methylation, but we methylate our DNA both naturally, and also we smoke cigarettes sometimes. That can sometimes add modifications to our DNA that we may or may not want. So our DNA is being modified all the time. And that's actually one of the key things in cancer, because in cancer, our methylation status changes. So we can actually classify, stratify different kinds of cancers by the different kind of methylation that you see within those cells. So every cell may contain differently methylated DNA. So that's true, but this is the exciting thing. We can literally look at the living representation of a cell's genome by looking at methylation. It's probably far more representative of some of the activity in the cell than just reading the bases themselves, which is the same in every cell.
Well, I think with the launch of the GRAIL methylation test, which has been rolled out to 140,000 patients, that's bisulfite methylation, I think we're going to be hearing a lot more about methylation in the coming years, pan-tumour.
Bisulfite methylation is pretty expensive. You get methylation for free with nanopore sequencing. So that's worth remembering.
Okay, so there's really, really a lot of promise out there. Yes. Good. Is there anything else that you would like to say about what is happening in your lab and what you're excited about beyond nanopore sequencing?
I think it's the ability to rapidly achieve patient benefit. I mean, short-read sequencing is great, but there's always a but. It needs a bit of time to batch enough people to get it going to make it cost-effective. With nanopore sequencing, we can turn genomes around really, really rapidly, 24 hours or less, depending on what the application is. Something like methylation, you don't need a high read-depth coverage, you just need enough to do the methylation call. And so by using this technology, we've been able to access samples much quicker and get much more information. The data is fantastically useful. I think the other thing that's most exciting about nanopore, it must be about the only technology in sequencing where when a better base caller comes along, you can reanalyze the data set you've got and get more accuracy and more insights. Retrain, like Greg talked about base modifications. One of the things we're working on at the moment is developing a better base modification caller. So we can access some of these base modifications that have been out of range of conventional technologies like hydroxymethylation in the future. So I think this is a technology that's got a long way to go in that there's a lot of stuff we could do with it we haven't been able to do before, and that's very exciting.
Again, one last question from the floor, and I think that's all we're going to have time for. This is a great question. Something that we're not very involved with yet in Genomics England is cell-free circulating tumour DNA. And of course, it would be incredibly valuable to take a long read or even a short read whole genome sequence from circulating tumour DNA. But is that achievable? Are these short fragments of DNA sequenceable?
I'm going to give away the trade secrets now, but I suppose people won't mind. So one of the really exciting things about the latest iteration of nanopore technology is the library preparation kit. So I'm sorry if the audience is going to get very technical, going to accept a very low input, femtomolar inputs, which means when you've got short DNA fragments of sort of 100 to 150 base pair long, that is compatible with putting it straight into the kit. So it is possible to input ctDNA directly into a nanopore reaction. Now, I know sequencing, it does require optimization, obviously, and there's a long way to go, but the technology is expanding that, yes, we are actually, we've done it, getting ctDNA onto nanopore technology just by direct ligation. So yes is the answer.
Fantastic. Bring on femtomolar samples. Exciting. Okay, well, thank you very much to both of you. And can I please encourage the audience, you can see the quality of the answers you can get on technical, but also non-technical questions. So please do ask your questions. I've got these coming up live in front of me right now. And if you two would kindly leave the stage, I'm going to bring on my next panel. Welcome. I'm very proud to invite to the stage Darren Trina, who's one of the leading pathologists in the country and perhaps our very own pathologist from within Genomics England. Please can you introduce yourself a little bit more fully than that, Darren?
Thanks, Parker. Yes, I'm Darren Trainer, a pathologist in Leeds. I'm one of the doctors who diagnoses cancer in the lab. But my other job is to be the director of the digital pathology program called NPIC, National Pathology Imaging Cooperative.
And how many hospitals are in your NPIC?
It's pretty big, so the ambition is a national central system for digital pathology to which other scanners can be added at other sites. We have at least 20, possibly 30 or 40 hospitals by the end of the programme.
That's quite an achievement. Perhaps, please introduce yourself.
Hi, so my name is Pablo Morgan. I was a pathologist, and I'm now one of the directors here at Genomics England, and I head up the multimodal imaging programme.
Perhaps you'll always be a pathologist. So, Darren, can we first just start clinically? You've been one of the leading lights, not just nationally, but globally in digital pathology. I think you introduced it in the UK far ahead of other UK hospitals, but also international hospitals. Perhaps you can tell us what digital pathology is doing for our patients and doing for the operational workings of cancer diagnosis in the clinic today.
Sure. So it's a really very simple technology. Digital pathology is a technology that has taken its time to come, and that's the reason being is because the images that we take down the microscope are enormous. So we pathologists have been working for decades using microscopes to look at patient samples on glass slides. And what the scanners that we use in digital pathology allow us to do is fully digitize that entire little piece of glass. So a single patient's sample might be maybe a centimetre or two in size on our glass slide, but when we scan it, it creates an image the size of a squash court. And that is a lot of data. That's the reason why digital pathology has taken a little bit of time to come to the mainstream. It has a variety of benefits of digitizing our samples in pathology labs. The most obvious ones are that we're no longer tied to that physical glass slide anymore. So we can review it in different rooms, in different hospitals, in different regions or even different countries. And that really opens up lots of new ways for us to work in pathology. We can get second opinions more rapidly. We're not relying on samples to be sent in the post to other laboratories for their opinions. We can collaborate more readily with larger groups of people. And then in this era of artificial intelligence, having the images digitized means that our images are available for image analysis for using AI, which is a very exciting new area for us.
So we're going to come on to AI, but I just want to go, I've done a tour of your lab, which is really inspiring. And when you mentioned an image the size of a squash court, you weren't actually being metaphorical. Can you describe for us what you've built and how that helps you in an MDT session?
Yeah, so because our microscope images are taken at such high resolution, the typical scanner that you might have in an office is about 1,000 dots per inch to generate an image. The scanners we use in our pathology labs are 200,000 dots per inch, much, much higher resolution. So as a result, each image file from each patient is very large. And some of the early work that we did at Leeds was to evaluate, well, if you're looking at such a big image, what's the best way to do that? Because it's a hard thing to do with the computer. So we eventually decided that what we needed was very large displays with many megapixels or tens of megapixels, initially experimenting with a wall of displays which had 50 megapixels in size, and we found that was a really good way of interacting with the images. As time has gone on and computers have got faster and displays have got better, we found that it's very possible to essentially replicate the microscope diagnosis on a desktop display, which is a moderately high resolution today.
That's really impressive to see. So you've been speaking about really the clinical implications of digital pathology, but perhaps what we're working on in Genomics England is the research potential of not just digital pathology itself, but combining that with the molecular modalities and also the clinical data. Maybe you could just describe the program that you're responsible for.
Yeah, okay. So we're incredibly privileged at Genomics England because our consent is quite broad, so our participants are very generous in terms of the fact that we're allowed to access not only the data from their genome, but we can also access clinical data about those participants and also now the images. So what we're trying to do is link all these genomes, the images, and effectively the digital pathology and radiology for all of these genomes. The focus for us is cancer, so we're working on the idea that there's about 15,500 what we call solid tumours. So we're not really looking at hematological or blood tumours, and we're trying to digitise all the images that we have for all of those participants. So at the moment, that is very much a logistical process. So we're going back to each NHS laboratory, asking for the physical glass slides and then sending them up to Darren and his team in Leeds, who are digitising those images for us. And then we're also doing the same with each of the NHS sites to retrieve the radiology image as well. So the first part of our project is effectively creating a very large data asset or a library.
And before you go on to the second part of the project, can you be clear, are we just collecting the index slide, or what are we collecting in the pathology setting?
Yeah, so the 100,000 project was slightly complex in terms of we created a tissue pathway. So we had the fresh frozen section, which we call the index slide. And then that participant would then go on and have what we call the standard of care. So they would have their full specimen resected and excised and create FFP blocks. So we're actually going to retrieve the index slide and the whole excision specimen. So for instance, in the colorectal tumour, we'll have probably about 15 or 20 glass slides per participant for that excision, and we're going to do that for all of our participants. So the library and the image library that we'll have potentially could be one of the largest libraries in the world of linked images to genomic data.
And clinical data. So we will really understand the full spatial context of the tumour.
That would be the aim, Parker. I think the problem is obviously at the moment no one's really had this amount of data, and that's always been the challenge, is actually having access to this kind of data. And I think because of the privileged position that we have, we're able to do that. So we can provide researchers access to images, to genomes, to the data to actually try and work out what these changes mean, what do these mutations that we've seen in the genomes that we've got a sense and an understanding from, what does that translate to? So can we really see, an ER change from a kind of a genome? Can we see that an H&E stain on a glass slide? And can we see more? What can we understand from, survival, drug discovery? And I think that's where we need to go to next, really.
Okay, and then the second part of the program, which is very much about bringing machine learning tools to the image, what are we doing there?
So Genomics England at the moment have what we call a trusted research environment, or TRE, and at the moment we keep all of our genomes and our clinical data within that, and we invite users to kind of what we call a reading library. So you enter our research environment and you review the genomes and the clinical data, and we will also add in the images into that research environment. But what we'd also like to do is adding tools to try and interpret the images alongside the genome. So what we'd like to do is provide researchers access to, say, 2,500 breast genomes, their comparative excision specimens, so that could be 10, 15,000 images, and the tooling to try and understand where the tumour is, where the stroma is, where the kind of infiltrating cells are, what that means in terms of the mutational changes alongside those what we call morphological changes down an H&E or on an image.
And I know from conversations that both you and I have had with the machine learning community that it's increasingly possible to vectorize both the spatial and the molecular context into a single vector and run that through a neural network and really get an unsupervised view of the impact of those multimodal features on outcomes such as survival and prognosis in response to treatment. So I'd love to ask you about that, Darren. What are you seeing out there?
Yeah, I mean, that's really exciting. I think the benefit of having the pathologist in the loop, so to speak, and having the images is that we can actually close that loop and make sense of it. So when you come up with predictions about survival or about maybe newly discovered subtypes of cancer, you can say, well, is there something to be seen when you go back to the images? We've seen early examples that are already in colorectal cancer, where one group has described a different prognostic subgroup within colorectal cancer, and then when they worked with the pathologist, they found some morphological features down the microscope or on a digital image that were associated with that. So it's a very exciting time. We can even start with simpler things though, so simply just confirming with an expert pathologist that this is cancer and that there may be some pre-invasive cancer alongside it. Cancers look different down the microscope, so when we take maybe 5 or 6 samples of a tumour, it's often surprising how different it can look from place to place, so we can inform a little bit of the understanding of the genomic profile that comes out of that. And then simple tools for calculating, how much DNA might I get out of this? Is this viable tumour? Is it necrotic tumour? Really simple basic things that we can provide with DigPath. And then, as Pablo said, we then have this really unique, globally unique data set that allows us to build on that in all sorts of ways using AI in future.
So in such deep specialisms, there's a risk of people working in a siloed way. Do you think that having the data from the genome and the image integrated will actually help interworking between geneticists and pathologists?
Yeah, good point. Yeah, I think it's essential, really. And, when I think we're only just at the beginning of this field. It feels like there's a new crossover field that needs to be emerging. We have a fellow that's going to be working with Genomics England, Charlotte Jennings, and her initial work for us is just sort of discovering what work has been done in pathology genomic correlation in the past and where the opportunities might lie. So even mapping that out is an interesting task in its own right. I hope to see a generation of clinician scientists working in this area following that sort of pathway. So yes, I think communication, I think involving pathologists in these discoveries is very exciting as well. So it's really good to see for the first time in a long time pathology images in Nature Medicine papers on a regular basis now because of this amazing new technology and simple technology of digital pathology. Hopefully it will improve the quality of cancer research by having that additional multidisciplinary view on things.
And a question for both of you. I'm seeing more and more exciting papers in increasingly credible journals that suggest that you can identify genomic features, genomic phenotypes in tissue, like HER2, like macrosatellite instability, for example. So do you think that the pathologists are going to put the geneticists out of business one day?
That's often a question posed when AI is sort of showing these superhuman abilities. I think we take a more real-world, nuanced view on it. So some of those discoveries are really very exciting. Some of them are based on kind of hunches that good pathologists would have had already, that, as I said, tumours can look different from sample to sample. So some of them are confirming that we knew. Some of them are really exciting new findings that we would like to see verified in larger settings with more diverse data. Perhaps the short-term value of it will be in helping us to excitingly discover new subtypes, new prognostic subgroups within cancer, but also just simple workflow improvements. So perhaps rather than putting the geneticist out of a job or not doing an assay, we might see it as maybe ordering the right test a bit quicker. Yeah. And we've seen 2 groups demonstrating that in prostate cancer already, where a H&E-based AI is looking at the original biopsy, is deciding whether or not it should order some immunostains, and it's saving a few days, 4 or 5 days on the prostate cancer diagnostic pathway for those patients, which is really valuable. So it's I think the real world is more nuanced and complicated, but in some ways better because we do it that way.
Well, you mentioned hunches, but hunches lead to anecdotes, but it's only when you do something at a national scale that you can convert those anecdotes into evidence base, and that's what we're trying to do, really to industrialize the good work that you and your colleagues in pathology and geneticists have been doing for many years. So I do have a bunch of questions that have come in from the floor, in no particular order. Darren, this is another one for you. This is actually a bit of an infrastructure question. So what are your thoughts on centralized storage of images and how best to ensure collaborative learnings from algorithms?
Yeah, that may come from a pathologist, I'm not sure. But yeah, we, on the NPIC programme, National Pathology Imaging Cooperative, we took a very strong view that there was an opportunity for the NHS by centralising. So we are building and will have a very large central data store available for any hospital to plug a scanner into. And the reason for that, a variety of reasons. You get economies of scale, you get better contracts by doing that, you can have an expert team running it. But in terms of AI, there are two key advantages. One is that you can then gather data in a structured, standardized way across multiple sites. And an argument, a failing of AI at the moment is that we're just not gathering broad enough data sets, particularly from non-teaching hospital settings. And then by having a centralized infrastructure, you can deploy AI much more easily, potentially at the flick of a switch by installing code. So with an NPIC, what we envisage is that once we've got to the point where we want to evaluate an AI algorithm in the real world, we install it on that central system, and that allows it to be used in all of the hospitals that are using our system. And perhaps as importantly, we'll have the staff centrally who'll understand AI deployment and evaluation, so that that burden of understanding that is taken off some of the hospitals using the AI. It becomes much easier to deploy. So a bit of a long-winded, complicated answer. But
it's a hard question. Thank you. I've got a question for you, perhaps. So this question is, are we only focusing on immunohistochemistry? And I can answer that quickly by saying, no, we're focusing on H&E images, which is the bulk of our collection. But why don't we try to address this question? Why are we not doing immunohistochemistry, and what is the challenge of centralizing IHC images?
We're not saying we're not doing IHC. So at the moment, the focus and what we have funding for is for H&E slides. We will also ask most of the NHS sites as we retrieve for the immunohistochemistry, but the problem is that will also include multiple levels, multiple sections, and actually the number of slides we potentially could have could almost, go up 50%, and actually that puts an impact on our storage, things like that. So we're not saying no to that. I think within reason we are asking for immunohistochemistry slides, because actually I think there is a real value to having the immunohistochemistry. So if you have an ER expressing stain, you compare that to the genomic markers as well. I think actually that could be very useful to do. So I'm not saying no to it. I'm just saying within reason, as long as we can do that, we will do that.
And clearly many of the driver proteins show up in IHC. So Dan, what are some of the challenges? Because immunohistochemistry is less standardized than H&E. So operationally, do you think that could be a challenge for us to make sense of kind of decentralized data?
No, I don't think so. I think it's quite doable. It's easier to image in some ways, yeah. I mean, ironically, when we started in digital pathology 20 years ago, the only image analysis that you could do was based on immunohistochemistry, because the images are very simple brown or not brown images, and deep learning has changed that. So as a result, all the image analysis now we see is on H&E stains. So I'd be pretty optimistic about including IHC. Yes, there are challenges with every lab having their own protocol and the results not necessarily being transferable. But if you need to do some just discovery-based work or comparing H&E with IHC, I think that the data that's in the 100,000 genomes data set is probably of some value. And I think we should make the most of it if we can.
And that optimism is why you're running National Digital Pathology. So a question for both of you from the floor. Can digital pathology images also be taken of metastatic lesions, solid metastatic lesions? And what's the opportunity there for longitudinal digital pathology?
So at the moment, we're only consented for what the participant from 100,000 Genomes project has consented us to take a sample for. So at the moment, that would probably be their primary tumour. If it is their metastatic lesion, then yes. The aim and the dream would be is that you have the primary diagnostic tumour. You then also have a digital image from the metastatic lesion if, unfortunately, they have a further lesion. That would be our aim. If you can then also relate that to multiple time course of genomes and then radiology, you suddenly got an asset that will be incredibly unique and incredibly kind of data driven that I don't think no one will ever be able to have. So it would be ideal, but at the moment we're only going on what we have consent for.
Yeah, okay. So I'm going to switch focus entirely now to a kind of operational set of questions. I think it's a tough time for the pathology industry. We have a massive shortage of pathologists. Maybe you can talk about that. And COVID has been very difficult as well. But maybe you can, Darren, you can talk about how digital pathology has changed, not just the logistics of the discipline, but also the lifestyle for new recruits and students.
Yeah, and I think for pathologists that are listening in, it's probably worth hearing that, yeah. So the places that led the world in the initial days of digital pathology when it was first developed were those that really had acute shortages of pathologists, in Sweden, northern parts of Canada, where they really wanted to try just keeping a service running. It's coming a little bit later to the NHS and to the US as well. But absolutely, I think one of my colleagues, Bethany Williams at Leeds, has written a very comprehensive review of the reasons why you might want to go digital. And there are all sorts of strategic reasons for the NHS, operational reasons for hospitals and laboratories, and individual reasons for pathologists. And if you just focus on the pathologists, lots of compelling reasons. I don't know whether perhaps it's developed a sore neck yet, but most pathologists have got some sort of musculoskeletal problems due to being hunted over a microscope for most of their life. Pathology is a job that sometimes is chosen because of the flexibility of the career offerings, and I think digital pathology has definitely enhanced that. So at Leeds, we've had 3 trainees joining our program out of 9 1 year who were coming because of the technology. We've had 1 consultant who now works a significant amount remotely from home, which completely enables their life to continue, their working life to continue. And we've obviously during the pandemic been very fortunate that we've been able to keep looking at patients' images and showing them at MDT meetings, and our trainees have continued to be able to look at images and be trained when times were such that you couldn't sit beside each other at a multi-headed microscope. So lots of key benefits, and I think that young pathologists coming into the profession see that. It's not an overnight switch from analog to digital in pathology. It's going to be a multi-year program, but I'm hoping that it'll attract people into the profession, make them feel they're doing something exciting and developmental. And perhaps we should say that they're not going to have their job replaced by AI now today. It's going to be very exciting because they'll be using AI to do some really cool stuff in the future.
Yeah, I completely agree. It's not man against the machine. And perhaps so far you've spoken mostly about what we're doing with our retrospective pathology data, but could you also clarify prospectively how we're going to support the NHS with prospective sequencing of patients in the GMS? And then maybe Darren could go on to tell us a little bit about the NHS's prospective plans.
Yeah, so the Genomic Medicine Service, at the moment we have a series of cancers that we're kind of standard care commissioners, and we can, those participants that have a research consent again, we will be making requests in a similar way for those images as well. So we'll ask the sites that will have the glass slides to digitise them. Some of them may have scanners, and so we'll be asking for the digital scans for those participants as well as their radiology images. So the idea would be is almost near real time as that sample is being processed, we'll be also able to digitise those images as well. And that probably feeds quite nicely into Darren and his national strategy.
Yeah, so I think we've been working closely with Genomics England for a few years now and saw an opportunity to align our vision with what you're doing. So as part of our ongoing funding of NPIC, we propose to develop 2 national networks, one in paediatric tumours and one in soft tissue and bone tumours. These are led by Neil Sabir in Great Ormond Street and Adrian Flanagan at Royal National Orthopaedic Hospital. And through those 2 programmes, we have a really very engaged community of pathologists who want to be part of that, and our funding allows us to put scanners into a lot of those sites to enable synchronous gathering of the digital pathology data alongside the genomic sequencing that's being done. It also brings other benefits to those communities. So Adrienne and her colleagues, only 5 of them in the UK are able to diagnose bone tumours or are certified to do it. So it enables that very small community to work together, get the right diagnosis quicker for patients. So we envisage national MDT meetings discussing patients, collaborating amongst that pathology community. in addition, so there are operational benefits in addition to the strategic benefits of scanning the digital pathology at the same time as the sequence is being.
So we can have a real-time MDT for example, an osteosarcoma, which is such a specialism and such a horrific disease.
Yeah,
I mean,
I'm quite excited about
that's
that.
just wonderful. And one thing I'd like to draw attention to, which I've read many times over the years, is that you did an extensive business case for why it makes sense economically for a hospital to introduce digital pathology. I'm sure you were anticipating many of the lifestyle benefits that then became essential during COVID, and you've made that available on your website. I know that there are a lot of startups and other companies that have benefited from that. Maybe you could tell us a little bit about how the economics of this works in the hospital.
Yeah, and I think at Leeds we're always very cautious about the technology not being mooted as simply a cost-saving device or a centralization of pathology labs device. So if you read the couple of papers that, again, Bethany Williams wrote about the future of pathology and how we could support it with digital, it's very nuanced. So yes, there are settings in which it's mandatory to have digital pathology because you've got a remote laboratory, you've got remote pathologists. But as I said, the benefits are much more complicated than that at different levels of the health service. And it also depends on the health service's willingness to deliver services digitally or otherwise. I think digital pathology in the NHS has definitely hit the tipping point now. So people are coming to us saying, we are going to go digital, tell us how to do it. Whereas 3 years ago it was, well, tell me why I should go digital. So the case for needing to be accepting of it is being made already. It will take time to establish that it's not going to overnight replace the traditional microscope and paper processes that we have. But what you find is that once you've gone digital, the benefits start multiplying, like all digital transformations. They take some years to come, but once you've done it, you couldn't possibly go back. And I think that's what we're finding so far. This is one of my jobs, is to encourage both the pathologists and the laboratories to consider the move to digital and support them in doing it, but also to influence those who are making big decisions, that they should be supporting the transition to digital, both financially and emotionally, I suppose.
Yeah, absolutely. And perhaps I'd like to ask you a little bit more about the machine learning program. You're not a machine learning expert yourself, but you are trying to orchestrate the coming together of this platform and tool set that will really allow different modalities of research on genomes and the spatial context. Maybe you could tell us a little bit more about how that program is going and what you envisage the research experience to be like once the product is built.
I mean, the sky's the limit, so I'd like to say the research experience will be phenomenal, and there'll be no questions about that. But I think at the moment we're working with users, so we need to understand this is ultimately going to be a very novel platform. This has never really been done at scale. No one's ever handled this much data, so we need to really understand what are the questions our researchers want to ask and how is that going to be done. So is it going to be very much a code-centric front end? Is it going to be point-and-click interface? And how much kind of input do we need to help them with that? The tooling is ultimately out there, like if you just want to look at a whole image and just identify tumour versus non-tumour, those tools, those modules, whatever you want to call them, already exist. But I think the challenge we now face is kind of integrating, this is tumour, this is probably an ER positive tumour, or this is a HER2 negative tumour, and what does that morphologically look like down a nuclei is actually a combination of a series of different kind of analysis tools. And that's what we're looking to try and bring in together. So it's all going to be ultimately a cloud-based research environment. All the images are going to be cloud-based. I think the big issue, or potentially one of the biggest challenges, will be that kind of computation element. So we talk about the GPU usage. The storage isn't going to be too much challenge, but that compute and the need to analyze thousands and thousands of images and genomes together will be a real challenge for us. But I think actually AWS are being incredibly generous with their time and with their efforts to help us with that. And I think we can kind of work towards them in delivering that. And I think in the next 6 months will be really interesting. I think we're looking to do a few small pilots with a few small use cases with our users, and actually working with a group in Leeds to see if we can do some small cases in ovarian cancer. And I think ultimately we'll work towards what is best for us as users, and ultimately we have an asset that we need to be helping as many people as we can to learn from. I think that's a real focus here, that we have something that we can almost push the UK kind of research environment a few years forward with this asset. And so that's the way I see it, is that how can we help as many of our academics and our stakeholders as possible.
Yeah, I agree with everything you said there. And if I address the audience for a moment, so Genomics England's research strategy is very much to be an enabling platform for the global research community, but for both academics and for biotech and pharma to come and use our data. Now, this is a cutting-edge field, multimodal genomic and image analysis. And I think we have quite a clear understanding today, as Darren's explained, of the clinical benefits, and we can also see companion diagnostic benefits out there. But we know also that there are drug discovery benefits to this approach. We know that there's going to be kind of digital target ID being done. We know that there are more biomarkers out there that will help to classify, stratify clinical trials, and we know that we can reverse engineer these algorithms that predict response or predict prognosis and look for the drivers of response in the features of these combined genomes and images, and then use that as the starting point for new discovery programs. So I would ask the biotech companies and the pharma companies who are interested in that to reach out to us, because we are really co-creating this platform around your requirements, and we need to have those. This is not something we have in-house expertise on. So a call to industry: come and work with us and help us create the largest national scale multimodal research platform on the planet today. Good, thank you. I think we should call on our next panel. Thank you, Serena. Thank you, Alona. So I'm absolutely thrilled. I have two real experts on the stage today. I'm going to ask them to introduce them to you individually, but this topic is on bioanalytics. And Alona, maybe I can start with you. Please introduce yourself.
Hi, I'm Alona Sosinski, Scientific Director for Cancer at Genomics England.
And I'm Serena Nixonal. I am a CRUK Advanced Clinician Scientist in Cambridge. I'm also an Honorary Consultant Clinical Geneticist in Addenbrooke's.
So Serena and Alona are both very humble, but I need to just explain a little bit more. So Alona has handcrafted all of the clinically accredited cancer biopipelines and diagnostic tools that we use to create lists of pathogenic variants that go out to the genomic lab hubs, and has really innovated in partnership with the Dragon team in Alona, abilities to predict productive fusions from short reads, and it's incredible what you do for us. We're very lucky to have you in-house. And Serena has been an extensive user of Genomics England data. Serena recently won the Steiner Award for Cancer, which is a bit like winning the Field Medal in maths. And so we've been very, very lucky to have Serena, who's gone deep into our cancer data set and created mutational signatures and callers for her homologous recombination and mismatch repair, and a whole pipeline of new algorithms coming. So we're delighted to have you here. Thank you for coming down from Cambridge to join us today. So first question to both of you. This is a conceptual one. I'd just love to hear both of your vision for cancer diagnostics in the next 10 years. Maybe we'll start in-house. Start with you, Alona.
Thank you, Parker, for this nice introduction. Yeah, it's quite a task to fit several years' vision into 2-3 minutes. I will try. Yeah, our current approach to the clinical interpretation of the genomic test is still very single mutation focused, and we are looking for the small variants, structural variants in a specific genes which can drive development of cancer. And at the moment we are working a lot with clinical scientists, with clinicians on supporting them in this journey. So we can have national platform which would support interpretation of the genomes, because genomes can be overwhelming. So we need platform which would help to sift through the thousands of the mutations, find the one which can drive cancer. So this is good. But these driver mutations, they can be missed sometimes, and sometimes they're very difficult to make interpretation of and find if they're pathogenic, not pathogenic. So this takes us to the next level of interpretation. If they do pathogenic, then they disrupt some functions, molecular pathways in the cell, and they leave big footprints in the genomes, hundreds and thousands of mutations with some specific patterns. So we want to recognize these patterns because many of them are linked to the patient response to treatment, to prognostic markers. So this is where the strengths of the whole genome sequencing come apart. And there is a lot of buzz now in a research community about these markers of genomic instability, but then the question is how we translate them from research space into the clinical space. And obviously, the easiest way is to do clinical trials using whole genome as prognostic markers, prognostic tools for these clinical trials. And thanks to the genomic medicine services, whole genomes are now covered for a lot of clinical indications. So we can develop here a new generation of clinical trials, which are based on the whole genome sequencing that used genomes for diagnostic of patient stratification. But so far, these patterns, they usually come from our previous knowledge of genome biology. And now we have probably enough data in our National Research Library to feed into the modern artificial intelligence, algorithm, deep natural learning, all this buzzword. You know. And until recently, we just didn't get enough biological data to use the whole strength. So now probably we are at the stage, and we mentioned before, like collaborations with Google and how we can use the strength of these algorithms to discover new patterns without previous knowledge. The next level is combine genomes with all multimodal data, and we probably already spoke enough about combinations with image data, biochemistry, knowledge about patient history. So to make it short, in, 10 years, artificial intelligence learned platform, which is trained from our multimodal data from the National Research Library. New patient arrive, you run it through this trained system. It extracts the most relevant bits of patient information from genomes, from images, from biochemistry, put it together into integrative markers for patient prognosis, whether the patient is likely to transform from primary to metastatic, what are the treatments that patients are likely to respond, and everything is presented into this interactive and intuitive way on molecular tumor board. So we support our molecular tumor boards in making decisions about patient treatment.
Wow, that's a very comprehensive response. Thank you, Aletta. Very good. Serena, how would you like to add to that?
And I hope that happens in our lifetimes. Yeah, I'm not sure there's a huge amount I can add to that, really. I suppose I could come at it from my perspective. We've been very fortunate to have had the opportunity to use the data that's been in the research environment. As you said earlier, over 17,000 whole genomes. That's the largest collection of whole genomes in the world. It is the most comprehensive data set worldwide, and it's been an enormous privilege for my team and I to be playing in the environment. And I think there's hundreds and hundreds of people who are doing the same. We've been sort of searching for patterns of mutagenesis. Alona's already mentioned them, these signatures, because we feel that they tell us something about the history of the cancer, how that cancer has arisen. And if you understand each person's cancer, then you might be better placed to treat that more efficiently. And so, we've, as you've mentioned already, we've come up with a couple of algorithms that help us, or help clinicians in the future, use genomic data in order to inform the treatment decisions going forward. This is only the surface that I'm talking about here, because as we've already heard from the other panels today, there's just a potential a huge amount of additional information that could come from digital pathology, from imaging that we could also gain from. But just from this little area, which is just somatic genomics, we've come up with a couple of algorithms that help us to look at whether a tumor is defective in a particular DNA repair pathway, for example. But just to give you one particular example, we did work in the lab. So, and there are plenty of scientists out there who are not data scientists who work in labs, and they'll find things. Now, if we all had access to the Genomics England data, it's nice and accessible, we could validate our findings very quickly. And in fact, what was mind-blowing for my team was that there was also clinical pathology information available. There was immunohistochemistry data there, and we were able to train our algorithm, validate it, and then publish it together with Genomics England, do a piece of IP together. And the whole idea then is to put it back into into the National Genomics pipeline so that everybody has access to it, not just us in Cambridge. You know, it is available to all the 7 genomic medicine services throughout the country. And, you know, it's just such a privilege to be a data scientist working in that system and environment and know that you can put it back into the national system for equitable access throughout. I think a really important point to make is that the insights we make are at population level. They're clinical or biological insights of a whole population, and it's anonymized data. And I think that's super important to realize. All of this is done in purely anonymized data, but the benefits are going to be felt at a patient level, at an individual level. And so I don't think people need to be afraid of their data being here in this environment. It's perfectly safe. I don't need to know who you are or where you live or anything about you. I just need to look at the genomic information and look at some of the clinical information in order to gain population level clinical insights in order to then apply for the rest of the population. So It's been a fabulous privilege to be working with Genomics England.
We're glad to have you working with our data. And I'm going to ask you a little bit more about the science in a minute and what a mutational signature is. But first, I would just like to ask, in the context of you being a clinician, you're not just a data scientist, and I know you've seen the impact of some of your algorithms and some of your work on actual patients in the clinic. So perhaps you could kind of lift this out of the detail for a minute and just tell us how patients are being affected by this.
So every genome is, really individual. Everybody's genome is just specific to them. And historically, we treat cancers as a collective. So, if you have an ER-positive tumor or triple-negative cancer, everybody more or less gets the same treatment. But individual patients may have something very specific wrong with their tumor, and then you'd want to treat that patient to the best of your ability. We still have a way to go before we can actually we'll need to go through accreditation, all those sorts of things, demonstrate prognostically that it can, it says something about outcome, demonstrate predictive properties. But we have had an impact on an individual, a couple of individual patients, for example, and that's been in the UK. So a patient in Hull, nowhere near Cambridge, who had a metastatic cancer for whom whole genome sequencing revealed something very specific about his tumor. And that meant that his tumor may have been sensitive to a particular drug. In this case, it was an immune checkpoint inhibitor, but he was metastatic. It's not licensed in the UK necessarily, so you have to, as a clinician, you'd have to go through trying to get IFR, individual funding requests, done. But in fact, it was turned down twice. So there's a piece of work that I think we need to do together with sort of legal frameworks and regulators in terms of how we can modernize how we use genomic data and data in Genomics England. But long story short, he got the treatment in the end, and it had a fantastic outcome for him. And you'd like to think that in our lifetimes, we will see, well, hopefully, within the next 5 or 10 years, we'll be able to get to a point where a genome sequence together with a pathology or radiology would help to inform decisions for individual patients as individuals, as opposed to as a sort of a collective bucket of diseases.
Yes, I completely agree. And perhaps you could talk a little bit about what you consider the role of Genomics England to be in the whole cancer pathway. There's no really other site in the world that has the centralized infrastructure that Genomics England has. It's done it in a remarkably short period of time, there's things that will need fine-tuning, but the infrastructure that has been built is pretty phenomenal, really, and I seriously hope that carries on. There's no other system that allows us to also recruit patients from all over the country as well, and be able to return then that information and that knowledge back all over the country. You'll need Alona's pipelines, you'll need input from Dragon, etc., to be able to get it better and better and better and return it back equitably. So I hope that Genomics England carries on with a sort of centralized way of being a place, or that data can come through, that also permits innovation, helps to develop some of the relationships with pharma, with regulators, to enable us to be able to do clinical research more effectively, and then hopefully develop the infrastructure, the interfaces that permit everybody to do clinical research, not just people with big IT teams. That just shouldn't be the case. It should be that anybody and everybody who's got questions, and there's lots of very smart kids coming
through the system, right, they need to be enabled to do clinical research. And hopefully that will come through Genomics England.
Yeah, I hope so too. I think we have made a lot of progress, and part of that is thanks to the push from scientists like you and Alona, but also part of it has been thanks to the pull from the NHS, who we just collaborate with and entirely depend on to drive this into clinical practice. And we are really impressed that even during these hard times for the health system with COVID, that the NHS is still progressing actually really quite rapidly and ambitiously, kind of driving genomics, not just whole genome sequencing, but also large panel genomics into the NHS pan-tumor. So Alona, one of the questions that just came in from the floor, which I think would be a good one from you, but feel free to answer as well, is about metastatic lesions and metastatic predictions. So really the question is, can we achieve with genomics that dream of thinking about cancer as a chess game where we can diagnose not just what is happening today in the body, but also predict future metastatic and future clonal evolution?
Yes.
Good.
Yes, I think I want to talk a little bit about real-world data that we managed to collect in our research environment, put it side by side with genomic data. We were very lucky in this country to have these centralized sources of patient clinical information, like patient electronic record. So we can put side by side genomes with patient diagnosis, histology, pathology reports. Well, they're still in the free format for many clinical indications, but still you can extract pieces of information from there. Information on patient treatment, information about patient mortality, some information about patient progression. At the beginning, there was a big lag between patient diagnosis and the time of this information arriving. Now we are probably down to 3-4 months with a lag time. So basically, we can follow up patients almost in real time, and for some of 100,000 genomes patients, we already have more than 5 years of follow-up. And we can see whether patient's disease transforms from primary to metastatic, or whether the patient is still alive, whether the patient is still free of disease after surgery. So we can start to put together this information with genomic information, compare cohorts of patients with transformed to the metastatic disease very quickly, patients which are still free of disease after 5 years, and start extracting these genomic markers which would help us to diagnose this patient, to prognose this patient when we see it at the diagnosis point, and predict whether the patient is likely to transform into metastatic disease, and when is it happening.
So I just want to add for the audience a little bit of context to what Alona said about real-world data. So we have integrated in near real time, I think we have about a 1-month lag with the cancer registries, which are hosted by, curated by Public Health England and hosted by NHS Digital. And what that's allowed us to do is really quite remarkable work with real-world data. So any of the listeners out there who are interested in this, an example would be a study that we did with PIK3CA, which I think is the second most prevalent oncogene. TP53 is the only more prevalent tumor suppressor gene out there. And it is a well-established fact, I think, it was published in 2012, that in breast cancer, the PIK3CA wild type ER positive, HER2 negative women conveyed a real survival benefit that you could see very clearly in the Kaplan-Meier curves. And we were able to recapitulate that with real-world data very, very accurately to a very low error margin. So there's just one example to show how real-world data that comes from the cancer registries, that's annotated from remission and relapse data that we have in our longitudinal data, can recapitulate RCT data, randomized controlled trial data, which is really, really very, very useful for people who want to retrospectively study subgroups.
Yeah, can I just add that on this recent American Association of Cancer Research conference, there was a lot of presentation around genomic instability, chromatripsis and how it can be, it can work as a predictive marker of the quick transformation into metastatic disease. So now we can use our genomic data, our real-world data, to confirm these findings.
Okay, thank you. We've got a question here that's come in. It's about tumour normal. So do you also sequence normal tissue to differentiate between rare germline and true somatic mutations with a matched tumour normal workflow, or do you rely on tumour-only variant calling? So I can quickly say we for every single cancer patient, we sequence the germline at 30x and the somatic tissue at 100x, and maybe you can both speak about why it's important to have tumour normal matching. Serena, do you want to lead with that?
So, whole genome sequencing has always been based on the premise of tumor normal matched sequencing, and yeah, their data is fantastic. High coverage. I think I don't think you do any tumor only sequencing, do you, in the?
Not at the moment. We are under a lot of pressure from hematological community to start sequencing tumor only for some acute leukemias, just because at the time of the diagnosis it's very difficult to collect germlines. We have a pilot with the skin biopsies, but it's not likely to work on the national scale. You're able to adopt to it in a big research hospitals, but probably not in every hospital. And of course, we want our service to be very inclusive. So we did quite a lot of progress in returning results for tumor only on hematological cancers, with expectation that once patient goes through the treatment, we will be supplied with germline as well, clean germline as well. So we will have both tumor and normal for research.
It's a good question, though. It actually shows what the potential is. So I've talked a lot about somatic mutations only, but there is a second genome there, which is the normal genome that anybody and everybody can use. And when that data is there, that's just the most powerful thing. And over time, it's only going to get better. It's only going to get more and more. Every time your data increases, your ability to learn something new just goes up and up. So you can do anything with it. Look at the germline more, look at the somatic bits more. So, the world is your oyster.
So size does matter.
It absolutely does for data.
I do feel that I need to ask this question because this question has come in from a patient. So this patient says, can those of us with metastatic cancer have our genomes altered to get rid of the cancer? Perhaps you could both talk about some of the strategies that are used for metastatic disease. We are not in the business of base editing genomes, but there are increasingly rich strategies for managing metastatic disease, and perhaps you can talk to that.
So as I understand it, you will be taking in patients who have got metastatic disease, isn't it, as part of your next phase. So I think we'll be learning a lot more about metastatic cancers. It's true that currently the vast majority of the data in Genomics England is on primary tumors, but I think that's going to change. There are certainly activities going on elsewhere in the world as well that are looking at whole genomes of primaries and of metastatic cancers. And actually all the data is very valuable, Genomics England together with all those other cohorts. But I think we're going to learn a lot more about metastatic cancers. As you say, we're not quite in the business of being able to change anything just yet. But yeah, I think we'll be learning a lot more.
And before I hand on to you, just for clarity, so about 70% of our cancer patients that we sequenced were sequenced treatment naive, so before treatment started, of the primary tumour, but they were stage 4 cancer patients, so likely already metastatic. Alona, do you want to make any comment about the metastatic setting?
Yes, I don't think we can edit genomes which are already affected by tumor disease, but we can develop targeted treatments which would only kill metastatic cells where we see these genome damages. Okay. So that would be the way of getting rid of metastatic tumors. Yeah.
A question's come in that I would like to answer myself, actually, and this one is, the question is, how does the wider healthcare professional or researcher access the gel data set? So I think this is a really important part to introduce trust and validation of the researchers that have access to our data. So we have a very, very rigorous way of managing who has access to the cancer data. First of all of our data, cancer and rare disease, lives in a trusted research environment, and it can never be removed at a patient level from that trusted research environment. It sits behind AWS's firewall, Amazon Web Services, which is extremely secure. They invest billions in security, and the information governance is very tight. To get access to the data, whether you're a researcher or a biotech or pharma company, you have to apply for access to our Access Review Committee. And the Access Review Committee is entirely independent of Genomics England. It's made up of professors and also participants who literally have skin in the game, who have their samples in our biobank. That Access Review Committee assesses, number one, is this the type of research, is this the type of company which is bona fide, which is qualified to have access to the data, and then they have to explicitly agree the research study that they choose, that they've written to study. And there's a technical summary and also a lay summary of every research proposal so that our participants can review and approve it. And then at the completion of the project, if they want to take aggregate results, so algorithms or GWAS results out of the research environment, that has to go through an airlock process. So our environment is not connected to the internet. The only way to get things out is a manual approval process by our airlock team, who literally review manually every single extract request from our environment and ensure that there's no kind of nefariously or accidentally re-identifiable patient data kind of hidden in those results. So it couldn't be more secure, and we're very, very grateful for our participants in making their data available for these research purposes. I hope you don't mind me covering that for you, but I think that's a really, really important thing to say. I've got a question that's come in. Do we have plans to extend genome sequencing to other ethnic groups? And perhaps Alona, you might want to talk a little bit about our diversity program. I can support with that if you want. And Serena, you can talk about the importance of diversity in genomic research.
Yes, of course, Parker. So this is the program that was recently launched in Genomics England, our diversity program. The expectation is that we will learn more about population frequency of the mutations that we rarely, or any type of variations that we rarely see in our collection of the 100,000 genomes. So it will inform our interpretation of, for example, tumor mutations, and do they look like the population-specific common mutations, or they're rare and they can be pathogenic causative of the cancers. So it's very important to have this background information on prevalence of the mutation that you see in the patient genome, prevalence in the background population. So this is probably one of the drivers.
Okay. And do you want to add to that at all?
No, I think it's really important for us to reach out to all the communities out there in the UK. A lot of research, I guess, has been rather focused on sort of a white Western population, but the UK is becoming increasingly diverse, so we need to learn more about across the populations as possible, and hopefully something like this, where we come to a point where everyone's tumors are going to be coming through gel, we will learn from the whole population.
We will, and actually in a panel I think coming up next, we have an explicit session all about diversity in our diversity program. A question that has just come in, and this one I think is for you, Serena, and it's great because it gives you a chance to talk about your mutational signatures. So the question is, can you give more detail about structural variation and that you can discover through your research? And perhaps you might start off by talking by explaining what a mutational signature is and how that allows you to look at genomic instability across the whole genome.
Okay, so as your cells, you start off from a single cell, and as you become human, your cells have to divide lots and lots of times, and you can make errors during the process of copying your DNA, and you can acquire mutations. And you will have heard about mutations from COVID, for example. Now, cancers have historically been believed to have been driven by these driver mutations, these few events that confer some kind of selective or growth advantage. 1, 5, maybe 10 driver mutations per cancer, and people are really focused on that. But actually, as your tumor develops, it acquires thousands and thousands of mutations, and sometimes hundreds of thousands of mutations. And those mutations aren't random. They actually tell you something about the biological processes that have been going awry as that cell has turned into a cancer cell. And so mutational signatures are these patterns, are the patterns of mutations that arise because things are going wrong in your cells. And that can be from anything. That can be from within your cells. You could have be producing something in your body that's damaging your DNA, or you could have a repair pathway that isn't working, or you could be a smoker that damages DNA. So things from outside the body as well as inside the body, all of it can leave these patterns that we call mutational signatures. And when I was at the Sanger Institute, that's where we came up with this concept of mutational signatures, and we showed that you can see it in cancers. Historically, and that was just technological, we started with single base changes because that was the easiest thing that we could call. And today, people are starting to look at indels and structural variation as well. So it is one of the things that's amazing about whole genome sequencing. It's that you can see all these structural big pieces of DNA that are swapped around or moved to other parts of the genome. There are patterns in them as well, and those patterns tell you something about the biological processes that have been going wrong. Sometimes those structural variations can produce a driver event, like we've heard already today, and fusions, for example. But sometimes they can produce patterns, and those patterns say that a repair pathway isn't working, for example. So that's how structural variations are starting to be used, I suppose, in whole genome sequencing, and actually already has clinical applications in some certain tumor types.
It does, and we're planning on incorporating your algorithms into our environment, which call microsatellite instability, or mismatch repair, I should say, the precursor of MSI, and also homologous recombination, which is really one of the major forms of DNA damage repair. And for those of you who haven't read Serena's paper on mutational signatures, it's quite stunning to see actually how beautiful some of these signatures are. You can see these very smooth melanoma signatures where you can see the damage comes from UV light fairly consistently, whereas in your colorectal cancer signatures, you can see those subgroups of mismatch repair and non-mismatch repair patients. So it's something which is, I think, really helps to visualize the heterogeneity and really the consequences of these mutational pathways on patients. Is that a fair description?
Yeah, absolutely. Always ask someone else to explain your research for you, because they always do it better.
Well, they're also very colourful, which is really, really nice. Okay, I have a very technical question, which has got your name on it, Alona. So here we go. Could tumour-only variant calling be used to investigate why double-agent genes act as oncogenes in some cancers and tumour suppressors in others?
Yeah, I suspect it's come from some research.
I would have thought so.
So as I explained, this scenario for this tumor-only pathway that we're developing now for the NHS and for genomic labs, we vision to process tumor only, mainly to return a small set of clinically relevant results back to the genomic lab, back to the oncologists. And then once patient is treated, we expect to receive germline sequenced coupled with the tumor and put proper tumor normal matched pair into the research environment. So if you're talking into the more research questions, then we would still stick to the proper tumor normal pair.
Okay, I think that's a good answer. I've got a question that very much has your name on it, Serena, because it's about breast cancer, which is a topic we talk about a lot with you in Genomics England. I mean, the gist of the question is, what are we going to do about curing breast cancer? I think this has come from a patient, and I would love it if we use that question to explain the insights that you found into DNA damage repair, and perhaps to address the biotech sector out there about the opportunities for addressing more responders to kind of BRCA and other pangenomic features.
So this is an enormous question, and certainly not one that any particular individual would be able to answer, and will require the input from the community and from the patients and from all the clinicians to be able to get to. So first, I think we'll need appropriate sort of infrastructure to permit us to be able to play with the data and the data that's there, and invite other people from outside, other researchers, other clinicians and biologists as well, right? We need people to ask the right questions of the data, and data scientists may not have all the questions, and that's why the interaction between all the parts of the community is very important. We'll need pharma input to be able to help us perhaps develop those studies, those trials, because playing with data is one thing. You develop an algorithm, that's something, but to make it clinically useful, you have to demonstrate that it does something either about outcome or that it predicts something. So to do that, you will need trials. You will need to put it in a context of a study. And so we need and actually we already have a sort of a setup, which is the Genomics England setup, but we need to be able to invite others, other clinicians, other researchers, other clinician scientists and pharma to engage in order to use this environment, to use the connections and the GMSs across the country in order to be able to recruit patients into appropriate studies that can be cascaded nationally. I know that sounds almost highly ambitious, but I don't think it's beyond the UK to be able to achieve something like that.
Yeah.
And I think over time, because this is the other thing, over time you'll be accumulating data on those cases and patients, and the knowledge you'll gain from that is massive. So we'll need infrastructure to be able to do that sort of thing. We'll need interaction from the community. We need infrastructure that permits analyses across sites as well. Sort of more federation analytics. You've already touched on companies like LifeBit that are in that space. I think we need to be able to do more of that sort of thing, because of course there are other data sets that have been created there's Interval, there's UK Biobank, there's the NIH Bioresources, huge numbers of TREs across the country. We need them to be better connected to enable people to do research more effectively. Just needs to be fast and quick and efficient. And I think nationally there is a drive towards trying to build up federated analytics. And then we need to have the infrastructure to permit people to explore the data a bit easier, because right now it's still a little bit limited to people who have IT teams. We need to have the ways of biologists and clinicians who can kind of go into an interface and kind of explore data and how they want to. So I think infrastructure, community interaction is terribly important. And we need buy-in from the whole country. And this is something that we should all be proud of, and we should all want to be part of and contribute to and recruit patients to, etc. So I think it's wonderful to be part of the innovative bit of discovery and finding new things. But we need to, if we want to put it back to patients, we need to give it enough time to consolidate as well. So give our GMSs, our genomic medicine services, our clinical scientists enough time to develop the workforce, train them, educate them, put in the infrastructure, get the accreditation, have the rigor, because of course doing something as an academic is quite different to doing something in the clinical context. But once we have that, we've put in the consolidation and we have the innovation infrastructure, the world is your oyster. Then in the UK you can really start to explore things. So in breast cancer, for example, 70% of breast cancers are cured, but there's still that 30% that we don't understand. How do we now find those 30%, use this infrastructure that's been developed across the country, interact with the right pharmaceutical companies, and try to understand the natural history of the disease, try to understand how to treat these patients better. I think it's kind of an ambitious sounding thing, but I think it's something that can be, that is achievable in the long term if you have the right things in place.
It is ambitious, but cancer is an aggressive disease and we need an aggressive response. And I think what these 3 sessions with 3 experts from the wet lab, from pathology, from molecular biology show the importance of is really the convening role that the NHS and the Genomics England have to bring scientists, to bring pharma companies, ultimately have the drugs that give the kind of clinical pull to push patients through clinical trials. And of course, what we can always depend upon is the courage of the patients who are prepared to put themselves out there and try new things, not just for themselves, but for the benefit of future patients that present like them. So I think at that point, I'd like to end the session and just thank you, Serena, you, Ilona, and all of the previous speakers for coming down to London to talk to us about cancer today. Thank you.
Thank you.